associate professor at MIT. Otto received his BS in computer and electrical engineering at the Polytechnic University of Ecuador. His master's in artificial intelligence from Utrecht University, and his PhD in theoretical biology also from Utrecht University. His main research focus is the ecology and evolution of natural microbial collectives. And today he'll be talking a little bit about that research that his lab has done, as well as sharing some of his personal path to his current position for the benefit of our aspiring young scientists in the RMI program. So I just wanted to add one more thing, and that's that the Pathways to Careers in Genomics talks are something that we do every quarter. Our objective is to bring in BIPOC scientists who can talk about both their experiences and share the great research that they're doing. This is an initiative of the Genomics Institute, Office of Diversity, and the Research Mentoring Internship Program, which is under the umbrella of that office. And we want to acknowledge gratefully our funding from the National Human Genome Research Institute. So with that, I will hand it over to Professor Cordero.
Thank you very much for the invitation, and it's really a pleasure to be here and to have this opportunity to interact with you guys. So I will start by telling you a little bit about myself, my trajectory, and if you have any questions as I'm telling you my story, please interrupt. I think that should be okay. And afterwards, I'll start talking about what's on the screen. Which obviously relates to the work my lab has been doing over the last 5 years, over five and a half years. So, so let's get started. So you may have probably inferred from my accent, I'm not from the US. I come from this tiny country. It's down there. It's called Ecuador. And you know, there is not much science, unfortunately, in Ecuador. It's one of those places that in Latin America that where science is not as developed as, for example, obviously Mexico, Brazil, Argentina, Uruguay, even Chile, definitely, Colombia, at least 20 years ahead of us. So when I was growing up, there wasn't really, I did not have a very clear vision at all of what it meant to be a scientist. But I did know that I like science, because my mother always bought me these books that I like to look at, even though I didn't understand them. So I had this sort of scale of values in my mind of what, you know, it's a good thing to be a scientist. Unfortunately, there is no clear pathway of how to do that in this country. So I went for the closest I could find, which was engineering. And I ended up going to computer science, which in retrospect was a great choice, because it gave me a flexibility to move to other fields. I think that's something common that happens with people that study mathematics or physics or computer science, that you have a flexibility to move to different areas, and that's indeed what happened. And so at that point, I mean, I guess I was a good student, and and I had in Ecuador, by the way, this is what the university looks like. I think still looks like pretty much like that. It's not bad. Inside a pretty big jungle, and this is close to the city where I grew up, Guayaquil. And I know it's a good university. It's just that they don't do research as we are used to seeing in the US or in Europe. But they have an intense academic regime and so on. So I thought I came out of this university with a good background. But like I was saying, there are no clear opportunities there to do research, except that I found one person that had a little research institute on artificial intelligence and things like that. We're talking about approximately year 2000, 20 years ago. And you know, I really liked that, and I think I did well. I had a little paper in computer science. They call these things proceedings. It's like a 1 or 2 page paper that was submitted to and accepted in a conference, and I went there to present. And I think that probably looked good in my CV, so I had the opportunity to to do my graduate school abroad. I went to the Netherlands. And so now we're talking about this jump from Ecuador to Holland, where I studied, initially did a master's in AI, and then I did my PhD in mathematical biology and bioinformatics. So that was my transition, my first transition towards biology.
Did you struggle with language barriers, or in Ecuador you learned English?
I learned English, but yeah, I'm sure it wasn't that great, but it was good enough to understand what people were saying, and also in Europe, I mean, it was also not their native language. Most people that I was interacting with were non-native speakers, so we all had our weird dialects. But yeah, I don't think that was a big barrier for me at that point, at least. And then, you know, with time and studying, my English became better. But an interesting thing that I had to say about this experience in Holland, which by the way was great. The Netherlands is a great place to study and to live and to work. Was that there were a lot of, well, when I was a master student, I was in a class of 70 people approximately. I was the only non-Dutch, which, you know, it was interesting. I wasn't thinking too much about it, but it was a bit of a challenge sometimes, but nothing too dramatic. Then in my PhD and outside the university, there were people from all over the world. And so I had still friends, colleagues at least from Zimbabwe, Japan, other parts of South America, everywhere in Europe, you name it. I mean, there were people from all over the world, except Americans. They tend not to travel to Europe to study. But anyway, I think that was a really great experience as well. And it's kind of different from what you would probably experience in the US as well. So even though we were all by definition minorities, this concept of a minority as you interpret it in the United States, and this struggle between minorities and the majorities and the representation is not something that I was really thinking about when I was a student there. And it's partly because we were all foreigners and we were happy to be there and learn from each other, you know. But we were not there to be sort of part of the Dutch society. If I had stayed there and tried to, you know, live my life in Holland long term, it would probably would have been a big issue, much bigger than it is in the US, because European countries are much more homogeneous. So I think the US is, with all these horrible things that we have heard in the last 4 years, it's a really welcoming country for non-Americans that come here to do the things that we are doing, science and research and so on, in my opinion, at least. And so after my PhD, so we're talking about now, I did a master's in Holland and a PhD, that's 6 years total. So combined is similar to the experience of a PhD in the US. I came to the US to MIT, where I joined the lab of Martin Poltz. And this was an environmental microbiology lab. And so it was the first time in my life that I was doing experiments with pipettes in the lab. And it was exciting. It went well, I think. And so that also shaped my career significantly. And a lot of the things we do now in my lab are strongly influenced by that experience I had as a postdoc at MIT in the US. Then, so as you're already seeing very clearly in this picture, there's a lot of zigzagging. I got, after 3.5 years as a postdoc, I started to look for opportunities to move on as a professor. And I got an European grant that is very prestigious in Europe. I mean, sort of equivalent to an NIH R01. And this allows you to set up your lab anywhere you want in Europe. And so I went there to ETH Zurich in Switzerland, and I started as an assistant professor. It was a very difficult experience at the beginning because I was used to the American system. I really liked the American system, academically at least. The fact that it's a flat system in general, no hierarchies. And so it was difficult for me in Switzerland. It took me a while to learn how to move myself there. But early, before I found how to sort of move myself in that environment, I had an offer to come back to MIT as a professor. So I took it, and I think that was a great decision. At the end, I ended up staying a year and a half in Switzerland, and it was also a great experience. And I had a lot of colleagues that I still interact with, people I met while I was there. But anyway, I came back finally to the US as a professor at MIT. And I have been a professor in the Department of Civil and Environmental Engineering, where I did my postdoc for the last 5.5 years. And I mean, I have a lot. I could really give I think a 2-hour talk just about my experiences in all this zigzagging. But that's pretty much the summary I had. So I don't know if you have any questions. Maybe you can ask now, and then we could devote to this maybe 5 minutes if you have any questions, and then we can start talking about science.
May I ask a question again?
Yeah, of course.
So you started off as studying technology. Did that, and eventually that kind of diversified into environmental science, microbiology. Was it all like a leveling out, like you learned along the way, kind of a build up plate that helped you, or you just?
It was almost 100% serendipity, I have to say. I'm sorry if that's not a very satisfying answer, because it would be nice to say I planned this whole thing, but it wasn't like that. And it was more like at every, there was a lot of uncertainty, I think in general there is, but initially when I was in Ecuador, I didn't know I could go to do graduate studies in Europe and then end up in MIT. I mean, I had no idea, and that would have sounded completely out of the question. But I chose whatever maximized my chances of taking these steps. And in that case, for example, computer science was, as I said at the beginning, I think the right choice, because it made it much easier to move to other fields. So when I went to Holland, initially I did a master's in AI, but then I moved into this mathematical biology, and the entry gate was computer science, because I was doing computational modeling of biological populations. I mean, I could program. I can still program. I'm very fluent with programming and computer science and all this stuff, and the statistics that are involved in genomics. I had this baseline, right? So that made it easier. So at every point, I was sort of trying to maximize the opportunities that I had. And later, it's okay, I'm finishing my PhD. What are the options? These European countries, they're so generous that they will give you a fellowship regardless of your nationality to go abroad. It's competitive, but you have to apply. I applied, I got it, I moved to the US. And so on, at every point, it's hard to predict. It's really hard to predict where you end up, but the best you can do is to look for, put yourself in a position that maximizes the paths that you can take.
Thank you.
Yeah. Anybody else wants to say something?
Other questions? RMI scholars, this is your time. Don't be shy. Looks like Ruby has a question. She says, do you think being free to move around has opened up more possibilities?
Oh, of course. Yeah. I mean, like, not only more possibilities, it's just, I'm a totally different person that I would have been if I had stayed in Ecuador. And I know this because I see my friends that stayed, and it's nothing bad with that, but it's just, I have met people from all over the world of all different, well, not all, but many different religions, backgrounds, etc., etc. It's just, it's like the same thing. If you grew up in a small town in the heart of the United States, and you never leave, then your views of the world are going to be a certain way that is different than if you go to whatever, New York or San Francisco. It's some similar experience, just at a global scale. So absolutely, the ability to move is a big thing. And for Ecuador, I'm still Ecuadorian. That's my only nationality. It's very annoying because I need visas to go anywhere. Even now, you would think this guy's a professor, he doesn't need. Yeah, so I'll get the visa, but I still need to ask to go anywhere, and it's a problem. Well, now I'm in a position that I don't think the visa will be denied, but being in Ecuador, it's very likely that that it will be denied. So yeah, that's a mobility is a big issue. It's a really big issue, I think.
Otto, and in regards to moving around, if there were three pieces of advice that you would give to folks who are interested in perhaps trying to establish themselves in different parts of the world, what would be three pieces of advice that you would give folks who are sort of starting on their paths and would like to take on opportunities in other places around the world?
Yeah, I mean, I don't know if I have three pieces of advice, but at least one. I don't know. But so I don't know if you're thinking, I mean, for this audience, I suppose everyone is living in the United States. I don't know if some of you are considering moving abroad. Normally, people, Americans or people that grew up in this country tend to focus on staying on American institutions. I can understand the logic of that. You want to be connected to a certain network and so on. But Europe is very welcoming, and in many places is for example, orders of magnitude cheaper than it is to study in the US, even for foreigners. The language can be an issue, but countries like Holland, Denmark, some places of Germany, not France, they do things in English. They have academic programs in English, and you know, it's a great place for science. So I think as I would encourage people in general to at least consider that option, and it's really good for the early stages of the academic career. Later, once you want to become a PI, then the fact that they're so hierarchical in Europe is a really big obstacle. So, I mean, I don't know if I have three pieces of advice, but at least that was one thing.
That's great. I have another question that came up in the chat from Lynn, which is sort of connected to this idea of your mobility. She wants to know what your challenges were. You kind of have addressed that, so I'm going to add a little something to that. And you know, Lynn says, what were your challenges when you moved without knowing anybody there? And my question is, to add on to that is, what is your support network, or how did you develop a support network in a completely new environment like that?
Yeah, that's a good question. You know, looking back, I mean, I think it's also my personality, but I don't dwell too much on the challenges. I have to say, I'm mostly thinking, you know, what's the next thing? How to move forward? And I don't know. It's just, and I think this is a personality issue. Other people are more affected by the challenge. But there were a few, and I can mention it. In terms of the support network, I honestly don't know. I think I'm really a nice guy, but I'm not very good at making friends. My wife can confirm that, and so I've heard from I have an Israeli friend who's also a scientist, and it's really close to me. He says that I'm like a street cat, and so because I don't have a clear support network. But yeah, I mean, I don't know. I think when I was a student in Holland, we had a community at least of international students that I felt that, I don't know if that was a proper support network, but at least there were other people that you're friends with, that you hang out with, and you can do things together. In terms of them, you know, take care of each other in case that's needed. In terms of the challenges, I mean, it's something that I guess I want to mention that is relevant for part of this audience at least, is coming from Latin America. There is a perception that we are disorganized, lazy, we're always late for things, and so on. I have to say, in my case, that perception wasn't so, it wasn't just prejudice. I remember fumbling things like crazy at the beginning. I remember somebody asked me a Dutch guy, he was trying to be nice to me and invited me to play squash with him. And I just didn't show up. And I remember thinking like, I don't want to go. And didn't call him or anything. I mean, like, this somehow seemed not normal for me in my brain when I was 20 years old, coming out of Ecuador. So I had to fight with myself, you know, to correct this behavior, but also to fight the perception that oh, here comes the Latino guy that is like, of course, late, or he's gonna drop the ball or something like that. And I think this is I can see this is an issue that that could affect you know, a lot of us with our background, even if you're very diligent, you know, there is that expectation, and that's a little bit of an issue that I think it's annoying, but we can, it's up to us, I guess, to make a better impression, and to show that we can deliver. I was in a class earlier in February before this whole mess with COVID in Brazil, where I interacted with a lot of students from South America, and I could see these issues coming up again. Everybody was late for the class, and there's this thing that you don't see, you know, and the issue is that most scientists that serve for us as reference are Anglo-Saxon or you know, Germans or Americans or English, and so these are our references, and this is they come with a certain personality. It's much more serious, and this is the stereotypical professorial attitude. It's actually also, it has a strong cultural, I think, influence. If most professors were from Italy or Greece or South America, then we would have a different perception of what it is to be an academic. And so anyway, I think that's an opportunity for us to change that perception, you know, like wearing these flowery shirts, for example, instead of bow ties. Anyway.
Great answer. Thank you.
Yeah.
Any other questions before we move on to the science part of the presentation?
Did professors urge you to go anywhere, or all your choices were on your own?
No, I made the choices on my own. Yeah, I normally don't. Yeah, when people give me advice, I normally try to look for the opposite option. I don't know why. So my advisor in Holland wanted me to stay, but I don't know, whatever. It was pretty much my own initiative. Yeah.
Thanks.
Thank you for all the great questions. Any last questions? You can always put them in the chat box if your mic isn't working or if you're feeling shy about it.
I have one question, piggybacking off of what we were talking about earlier about the stereotypes and Latinos and stuff like that. Yeah. But I'm mainly curious about, how did your family in Ecuador take the news that you were going to leave Ecuador, and how were you able to create that bridge between like what you were studying and have them understand how valuable it is? Because currently I'm facing that dilemma myself. So. Well,
that's a great question. I tell you, so this wasn't easy, but it was never an option, I think, not to go away. And I think, so my situation is that I am an only child, and my parents divorced when I was 2 years old. So I grew up with my mom, only in a tiny apartment in Guayaquil. And but she's not the kind of person that would say like, you have to stay here, mijito, you know, and then live next to me. I mean, I think that would have been very nice. But so she always wanted me to make the best of my opportunities, and going abroad is clearly, when you are in Ecuador, the opportunity to go study in Europe is something that you cannot say no. You have to be a little bit crazy, I think. And you know, my mother luckily was very supportive and still very supportive, but it is very difficult because in my particular situation, she's alone, and she has been there alone now for 20 years. So managing this is a challenge, but I don't think, I mean, unless there are always special circumstances, but in general, I think families shouldn't hold back, shouldn't hold you back in terms of limiting your opportunities. And so, yeah, I mean, I don't know, I cannot say more specific things because it depends on each case, right? So that was my particular story.
I agree. Yeah, I think definitely it's a case-by-case thing, but great, thank you.
Thank you. That was a great question, Claudia. Yeah. All right, well, if there are no other questions, I mean, we might even have a little time at the end of the talk, so why don't we go ahead and move to part 2?
Yeah, let's do that. So here we go. So, okay. Let me make this small. All right, so we're going to start. So let me tell you a few of the goals for this talk. I mainly want to give you an overview, and I don't think time will allow me to give a full overview, but to whatever extent we can, I give you an overview of what we have done in the last 5.5 years, and you know, and what was the vision, and what were the challenges, and so on. So from the scientific point of view, the main sort of take-home messages I wanted to convey were this issue of microbiomes or microbial communities. This is the main subject of study of our lab. And I think there are a lot of fundamental questions that we should be asking about microbial communities. What even like the most basic questions, like what are these things? How do we define them? What are the units that define the microbiome? What is the structure of a microbiome, if we can even ask the question? So we like to move in that space of these general, hopefully important questions. And so when I say what are the general structural constraints of a microbiome interaction network, this sounds very abstract, and indeed it is abstract. But what I mean is, you know, we think that there's obviously a large number of different species in any microbiome. For instance, the gut microbiome, just to pick an easy example. But how do they interact? Is it a random network of interactions, or is there some structure? For example, a trophic structure, right? And don't pay attention for now. This axis is temporal dynamics. Let's forget about that for a moment. But simply, are these networks random, or is there something else that we can say that is more interesting about the structure of the system? You know, do we know this? Well, that's a good question. I'm not sure we know, or maybe we do. But let's talk about it later. Another question is, okay, if we imagine these microbiomes as networks of things that interact with each other, what are these things? Are they species? You know, I don't know very well your background, but if you're familiar with 16S ribosomal RNA or ribotypes, what are these units? Are they ribotypes? Or should we think about something a bit more coarse grain or more fine grain, strains? It's totally unknown, it's unclear. So that's another fundamental question, right? That we should be, I think, at least thinking about. And there is this preconception that because the mapping from species to functions, whatever the metabolic functions of the organisms are, is not a one-to-one mapping, but a one-to-many mapping, meaning that there are many species that can perform the same metabolic function, perhaps with quantitative differences. Then perhaps the system should not be defined at the level of taxa, but at the level of functions. This is this idea, perhaps if you're familiar with this, of that taxonomy is not conserved, but the functions are conserved. People have made these claims, especially for human microbiomes. But you know, what does that mean is not really clear. How do we define the functional units? It's also not clear. So and so those are some of the basic, most abstract questions that I would like to address today, time permitting. Okay, this is what I mentioned before. How do we identify the units? That's actually a really big challenge, those functional units. Okay, so that was the most abstract set of slides. Now we're gonna things are gonna become more concrete, hopefully, as we move along. And so all of this was lacking a context. So what is the context? And in our case, for historical reasons, because of my work in my postdoc, I decided to focus on marine microbes. So let me tell you a little bit about this. And it turns out to be the case, by the way, that most of the developments in modern molecular microbiology started with marine microbes. And there are a few reasons for that. There has been traditionally a lot of funding from different sources going to marine microbiology. It impacts the whole planet, and the system is tractable, much more tractable than soils, much more tractable than human guts, poop, I should say, to be clear. Anyway, and so in the ocean, we can paint this extremely simplified picture that you're seeing here. It's my own sketches of a loop that's important for the carbon cycle. So there is CO2 in the atmosphere that is fixed by photosynthetic organisms, algae, or cyanobacteria in the ocean. And then this this carbon is converted to organic matter, nitrogen too, and eventually the organic matter is released, but it's not going to go directly back to CO2. It's still organic matter, just in the form of carcasses of animals, or fragments of dying phytoplankton cells, fecal pellets, polysaccharides that are excreted, etc., etc. All these things is complex, undefined melange of different forms of organic matter tend to aggregate in little particles. And if you go to the ocean and you take a sample and you look under the microscope, you may see some of these particles. They can be of sizes of a few tens of microns. And so these particles are hotspots for biological activity, right? So the particles themselves, if you're able to look under the microscope, they are not necessarily perfect spheres, but that's where my background as a theoretician comes in. We like to simplify things. So from now on, you will see a lot of spheres. And so what's interesting from an ecological perspective is that, as I was saying, these particles are hotspots of biological activity and scaffolds for the assembly of microbial communities. What I mean by that is that in case you didn't know, by the way, there are between 1 million and 10 million cells in most parts of the ocean. In some parts, it's 100,000 cells, but in most parts, it's around 1 million cells of bacteria per milliliter of seawater. That's a cubic centimeter. So that's a lot of cells, and they're floating around. And some of them, not all of them, actually, a small fraction, are specialized to attack these little particles. They're looking for the gradients, the smells. Then they swim towards them. They attach. They adhere to the particles. They start growing, and they form a well-defined, physically defined community that assembles on each of these patches. So that's a beautiful process from a point of view of ecology and theory and evolution too. And so that's kind of the thing where I thought, this is a really interesting arena where we can start to ask specific questions. For example, what is the role of microbial interactions in the assembly of these communities, and how do the interactions affect the functioning of the system, which is ultimately the turnover of the organic matter, the degradation of the organic matter. So that's pretty much what we have been doing over the last 5.5 years. So getting a bit more specific as we go on, this is how we did it. This slide, you will see a few of these stages, and what I'm trying to explain here is our experimental approach. So back in this idea came to me when I was still a postdoc, so must have been 2013, and I was thinking there were people around MIT that were thinking at that time, if we have all this sequence data from different human guts, then we can build these correlation networks, concurrence networks, people call them, where there are two species that coexist in one of the guts, but they exclude each other, others exclude each other, and then that probably is a manifestation of the interactions between species. And I thought to myself, this makes absolutely no sense, because the gut is there's a million of different microenvironments in there. It's more like from the point of view of the bacterium, it's probably like the whole planet. So any correlations that you will see there may not really capture any interaction. So I thought to myself, what we really need to do is to look at the scale that is relevant for microbes, and this scale is at most tens of microns. In terms of like the secretions, for example, how far will they diffuse? And if you have a neighbor or a neighboring cell that you can interact with, what is the length of the neighborhood? And we know now that the neighborhood is the order of tens of microns. And so that was kind of the inspiration. So I thought, okay, let's mimic these marine particles, which are probably the hotspots where all these interactions are unfolding. And so we do that with this stuff that you're looking at there. Is a hydrogel bead made of chitin. The dark things are magnetic cores. And so the neat thing of this is that you have this little particle. Obviously, we have millions of them, and then you can deploy them in an environment, later fish them back, and you will get bacteria attach them. And then many of these bacteria adhere in a specific manner. For example, this is chitin. Bacteria have chitin binding proteins, then they will attach to the surface, and we can fish them out. And in this way, by the way, we have been able to cultivate what I think are the best chitin degraders that have been ever cultivated. Not that that's something, you know, like a significant record, but it tells you of the power of the approach. And the first things we did was to characterize the dynamics of colonization of these particles. And we do that with genomics. And I sketch here these wavy things, because as you will see in a second, the dynamics are what follow what we call in ecology successions. So there is something that comes in and it facilitates the invasion of a different species and facilitates invasion of another one, blah, blah, and then you get these waves of colonization. Something that we also enjoy doing very much, and we still continue to do, is to cultivate, to build large culture collections of microbes. I really like doing this, I have to say. Cultivation is one of the things that has been, to some extent, forgotten because of all the amazing omics things that people can do these days. But there's nothing beats having the microbes alive in the lab, so you can study their physiology, their behavior, all the weird things that they do. I really think that's extremely valuable. And so we have culture collections now in the order of, I don't know, 1,500 strains in our freezers. A few hundred of them are genome sequence. And this is very powerful. And the reason why it's so powerful is because then we can, once we have the living creatures in the lab, we can phenotype them. So we can ask what do they do? That's the stage 4. And then once you figure out what they do, and I'll give you an example of what I mean by that, because phenotype is an undefined quantity, and we don't know how many dimensions it has or what. But anyway, once we have these phenotypes, then we can essentially map the phenotypes back to the community dynamics. You know, we can say, because we can map the isolates back to the culture-independent data, we can ask whatever these organisms do, for example, in the context of chitin, do they actually degrade chitin, yes or no? Is this happening, you know, in the early stages of colonization, in the late stages of colonization? That's what you can do because of this, the power of phenotyping. And finally, moving forward, the stage at which we are now is assembling these communities in the labs in synthetic ecologies to try to understand more about their interaction, dynamics, etc. That's pretty much the sort of the pipeline. All right, so here's example number 1 of how this pipeline works. So we did pretty much what I described before, with these synthetic particles, in this case made of agarose, alginate, chitin, carrageenan, different polysaccharides that are relevant in the ocean in particular. And you have the pictures there. That's not super exciting. It's just little beads. And the green things is bacteria that have adhered to the particle and are forming sort of little colonies. And they're stained with DNA stains. That's why you see them fluoresce. Anyway, over time, thank you very much. Over time, we can collect well, first of all, we do an incubation. We put these particles in seawater, just raw seawater. Close the bottle. We have it rotating overhead, and then we can take the samples every few hours just to see how the communities are changing on the particles. And when we take the samples, we can magnetically separate the particles from the seawater. All right, so, and this is a complicated picture, and it takes me a little while to explain. So maybe I'll explain these things, this type of pictures, and you tell me if it's not clear, because otherwise I'll be moving on and then you'll be wondering what is it that I was talking about. So these are heat maps. Okay, first statement, heat maps. And the columns represent time. These are samples taken at different time points. And the rows correspond to taxa, 16S, ribosomal RNA, amplicon sequence variants, if you know what that means. Essentially different versions of the 16, different types of sequences. And then for each time point, so that will be a column in this heat map, we have a different abundance of different taxa. And so obviously that's changing over time. And we have ordered the taxa by the average time of arrival, or the average time at which they became most abundant in the community. And so, and this heat map is normalized by rows. So that's why you see here, for example, I'm moving my mouse. Let's pick the first one, agarose. So there is something that appeared at the first time point that we sampled that taxa was there. Later it disappeared. It was just probably something that colonized passively. In the meantime, something else was coming in and stayed a bit longer. It became abundant and then disappeared. In the meantime, something else came, became abundant, disappeared, and so on. These are the waves of succession. Yeah, but remember the heat maps are normalized by row. So the community, as you go down the succession, the community is also growing inside. So if you don't normalize by row, you will only see the things at the very end. All right, so that's the explanation.
How did you track the heat maps? Did you basically take samples of bacteria and then phenotype them to identify, or no?
No. This is basically going back to this picture. This is taking samples at different time points. So let's say time, whatever, 24 hours. So we take these beads, separate them magnetically from seawater. We extract DNA, we send it for sequencing. And that would be, 24 hours is around here, right? In between 0 and 48. And so now once we sequence, we get a certain profile of the community composition. And that is what's here on the column. Basically, it's telling you that all these different taxa on top here were abundant, these different species were abundant.
Gotcha. Thank you so much.
Whereas if you come at 144 hours, it was different thing. It was whatever is down here. Yeah. Cool. Yeah. So that's the business, and that's how we detect these dynamics of succession. Yeah. And which I like to use this fancy terminology that I borrowed from chemists, multi-generational kinetics, but it's basically successions. Now there is this perhaps a little bit subtle question. He said, what we see here is basically the dynamics look very similar, right? These curves, these successional dynamics look pretty much the same between the 4 different substrates. I couldn't tell them apart, but I didn't explain this, but each of these heat maps is independent of each other. And with that, I mean that whatever was the species that was in any of these rows in heat map agarose, I don't even know if it is present in the other 4. The rows are not aligned at all. There are 4 independent heat maps. So it could be that all the species are different between the substrates. It could be that only a fraction of them is different. It could be that whatever showed up at early time points in whatever chitin showed up late in agarose, etc., etc. We have no idea about that. So that's the next set of slides, which also involves complicated pictures. Afterwards, it gets a bit easier, but it's the same type of heat map story. So that's why it's important to try to clarify this now. All right, and here we go. So it's the same type of heat map, except that now the rows are aligned between the substrates. So what that means is that this row here where I'm moving my mouse, whatever that species is the same one in agarose, and I cannot see. I think this is chitin, and it's the same one in carrageenan. Yeah. So what are we seeing here? What we're seeing here is that there's a bunch of species that seem to be very abundant, because in this case, as you see, the warm colors are proportional to the abundance. They seem to be very abundant, and they are present in all different particle types, independent of what was the substrate. They didn't care. And by contrast, if you go down here, let's see, for example, these guys here, many of them are present in other substrates, but they're particularly abundant on, I believe this is alginate. And these guys here are particularly abundant on carrageenan. And this down here, particularly abundant on chitin, and so on. The white lines that divide the heat maps, in particular the horizontal white lines, these are not put there by hand. We did a clustering of the species based on their profiles, on the temporal profiles in the different substrates. And it turns out these are the 5 clusters that you find. One big cluster for the species that did not care what the substrate was, they were always there, and 4 different ones for the specific, for the polysaccharide specific species. If there are any questions, maybe it's another good time to ask. Okay, so maybe I'm explaining this well. So what I have, forget about this. This is not going to tell us anything new, and this is just examples of the trajectories during the community assembly dynamics of species that belong to the different groups that I showed. So, for example, we call these species in the polysaccharide independent ones, the ones on top here, the ones that are present in all polysaccharides. You call them broad range. You look at their dynamics, the colors represent the different polysaccharides, the different primary substrates of the community. Remember that that's the main carbon source of the system is I'm saying main because there may be other things in seawater, but by far the most likely carbon source of the system is the polysaccharide in the particle. There's nothing else. And here are the dynamics on the different substrates, and as you can see, they're all very strongly correlated. So whatever they do, if they peak early or late, they were doing the same on the different substrates. They did not care. And then the narrow range ones, obviously appear only in one of the substrates, right? So then you see only one of the colors.
I have a question.
Yeah.
So why is there a bigger group for the non-specific polysaccharide species and a smaller group for the specific, well, narrow ones?
That's a good question, and I don't know what is the answer. I mean, I can speculate. There are a number of things, but the most important thing I can say is that this happens to be the case, and it's something that we should be aware of. It's one of the things we have discovered, that basically in all these communities, the actual things that degrade the material are in the minority. Most of the things that you have there are this substrate independent species, but I haven't told you yet what they do and why they are there. So maybe when we get to that slide, this will make a bit more sense. Yeah. So I believe that's, if not the next slide, comes after that. So this is I'll talk about it, but it's not super important. It's just showing, because I don't know if this slide was there, I think, for people that care about marine microbes. And you look at, and those people will look at these species names and they'll be like, aha, my favorite taxa. But if you don't know about it, perhaps it's not so important. Anyway, the most important thing I can say is that this is what you see down here is a phylogeny of the different types of species that we find attached to our particles. And as you can see, just from the different names, there are many different things, and in some cases different phyla, like Phleobacteria and Pteromonas are different phyla. And those groups that are specific to the polysaccharides are in red. They are like specific clades in the phylogeny. For example, I happen to know that this number 8 is a Psychromonas. It's a type of Pteromonas. For whatever reason, these things, they like to degrade alginate. And many of these organisms have a lot of enzymes to degrade alginate. And they, sure enough, they appear specifically on alginate and not on anything else. And so on. We can keep going. Whereas the other guys that are unspecific, they are distributed widely across the tree of life. Yeah. Now, here things start to get a little bit more, I don't know, juicy maybe, like addressing, I think, some of your questions. So let's go back to this example of chitin. And what you're seeing here is the same type of data I showed you before. Again, you have here a heat map, as I explained before. The rows are different taxa, species, if you will, if you want to call them like that. The horizontal axis is time. And again, black that means that the species was absent. And in this case, sorry for the inconsistency in the color coding, but red is the maximum abundance. Again, you see the succession, but why is this heat map so small? There are only 16 rows. It's because here I'm showing only those species for which we had a representative in our culture collection. We were able to culture something, and when we compare the 16S RNA sequence, it's an identical match. And then we know we have that thing that we saw in the culture independent data, we have it in the lab. So now, remember the pipeline, now we can do the phenotyping and then map the phenotypes to the culture independent data. And that's extremely powerful. And so, and this, the table here with black and blue, those are the phenotypes. So we basically, for each of these isolates that we have in the lab, we can ask the following questions, among many others. Do they grow on chitin, which is what the particle is made of? They should be growing on chitin, supposedly. That's what I expected, at least. If they don't grow on chitin, well, sorry, do they produce the chitinase, which is the enzyme that degrades chitin? That should be correlated with growing on chitin, we think. And can they grow on the monomers or dimers of chitin? These are the byproducts of hydrolysis. You know, like I was honestly expecting that most things would be either things that degrade chitin or they would be taking advantage of the degradation byproducts, akin to what people call cheaters in social evolution. And the last question we asked, are these bacterial motile? Turns out that, to our surprise, there was a very strong, as you can see, divide between the phenotypes of the early colonizers and the late colonizers. And now that I've told you that thing about the substrate specific and unspecific taxa, I can also tell you, as is written here, that the early colonizers, these are the chitin-specific degraders. You know, as you can see, they actually grow on chitin. They excrete the chitinase, that is the enzyme that breaks down chitin, and they grow on the monomers. Turns out to be the case, they are also motile. They can swim really fast when they detect gradients of these monomers, of these soluble monomers. And to our great surprise, everything else couldn't care less about chitin. So these were the substrate specific guys. But so what are they doing? Right? So that's the question. And that was our first surprise when we were doing this type of work. So here's something we did, and I'm jumping a little bit from substrate to substrate, but this was a follow-up study that we did with alginate, where we see essentially the same type of patterns, where we wanted to understand what's, oh, there is a question. Okay, hi, Fidnat. Tell us.
Great talk. I was just curious about if you control for attachment to chitin, right? Because if they cannot stay, they're not going to degrade.
Mm-hmm. Yeah.
I was just wondering, how are you controlling for that?
I don't know what you mean by control.
So if cells are not, are all of your your collection of bacteria, early, late colonizers, are they equally able to attach and stay on chitin surface?
Yeah, well, we collect them from the particles, right? So we separate the particles and then plate from the particles. So they were attached at least at some point. Now, how long do they stay attached? That's a really interesting question, and it's kind of a complicated answer. It is regulated, but in general, so this is going in a different direction, but in general, the residence time of degraders is surprisingly short. In our measurements, around 3 hours. And this is not because they cannot attach, it's because they like to leave.
They degraded.
And there's a story behind that. But whereas this we haven't measured it very well, I think, but my impression is that this unspecific taxa, that they actually like to stay there longer.
Great, thank you. Sorry to break.
Yeah. So I was going to say, I mean, here I showed you basically from the previous slide that there's this whole bunch of organisms that appear in the communities and are not doing anything that seems to be relevant for what the community should be doing, right? Degrading that polysaccharide, which was the main substrate. And so the question is, why are they there? What are they doing? And that's something we try to address in this experiment with alginate as a main polysaccharide. And the reason we use alginate was also because alginate can be dissolved. And so the experiments were easier. So we wanted to do metabolomics. So we took a degrader of alginate, we put it in media with alginate, it's the only carbon source, and sure enough, it degrades. And then we take the byproducts of its growth, so basically whatever metabolites are in the medium, we measure them, and then we feed them to the bacteria that come late to the game and that seem to not to care about the substrate, and to see if they can grow on that, on those metabolites.
Are the bacteria different sizes? And if so, does their presence affect the size of the particle affect how many bacteria could attach based on their mass and surface area they cover?
No, most of the bacteria in this case are around a micron, between half a micron and a micron. I don't think that makes a big difference, but I mean, encounter rates are a function of the radius. But I'm under the impression this is a quantitative question. I would have to look at the calculation, but I think at those scales that's not the main driver. It's more whether the cells are motile, for example. That really inflates the encounter rate significantly. And yeah, but I don't think this matters much for the things that I'm describing here, unless the particle was really huge.
Yeah. Thank you.
Yeah, and so I was saying here well, what you see there's two plots, right? So on the left you have the growth of the alginate degrader, and there's a bunch of other taxa, other species here that are in gray and orange. They cannot grow on alginate, so they are just down there, lingering, a very low OD, optical density, and you see the growth of the degrader, perfect. Where you see this dashed line, that's where we took a sample. We removed the cells and collected the media. This media has the metabolites that result from the growth of the organism. And we present this buffet to this other bacteria that cannot grow on alginate but appear to be abundant on the particles. These are many of them. If you care about these things, there are many alpha proteobacteria. They turn out to be the main sort of taxonomic group that shows up in this role. And there's a Vibrio there as well, and a Flebobacterium, and they all grow relatively well on the metabolites. For reference, the same organism that degraded alginate is here in the dashed red line, and as you can see, it cannot grow very well in its own metabolic byproducts. There are other organisms that are sort of specialized to take the metabolic byproducts of the degraders. And so that's a picture, and this is just a sketch that we built for the paper, showing some of the metabolites that we detected in this analysis. There are, in general, just a bunch of metabolic intermediates, nothing really very specific to mention there. TCA cycle intermediates, amino acids can also be excreted for a multitude of reasons. We also found the alginate oligosaccharides. Some of the organisms were able to consume them, and so overall, the picture here is that I'm trying to summarize in this slide, is that I think we are able to sort of reconstruct and simplify the logic of the assembly of the community. Instead of describing this whole process as a shopping list of species with their complicated Latin names, we can define it in a more functional manner. So here we have basically groups of species that are specific to the polysaccharide. You know, these are the degraders. These are the organisms that have the adaptations to adhere to the polysaccharide, secrete the enzymes, degrade, grow on it. And there are other groups of species which are there mainly as secondary consumers. That's a keyword. That's the way we like to call them. And they utilize the metabolic byproducts of the growth of the degraders. And these secondary consumers turns out to be the case. They are unspecific. So I call this like a core module. And if you want to assemble a community with polysaccharides, you just have to sort of connect. This is my computer science background, I guess. Connect the modules that allow you to eat that substrate. And that's actually the logic by which metabolic networks are designed. We have a core module in the metabolic network, for example, TCA cycle, the biomass producing reactions. And then there are these feeder pathways. If you want to eat, when I say you, I mean a bacterium. If you want to eat a different substrate, then you just get the transporter and the right pathway that connects it perhaps to glycolysis, and that's it, and so on. And I think it's the same logic, but at the level of communities. Any questions up to this point? No, good. I'm hesitating a little bit about how to prioritize things next, but you know, I'm going to skip some things that will follow here just to move to a different topic that I think it's a bit more related to genomics, and and kind of exciting. Something that we are about to submit. We have decided we'll submit this weekend. So anyway, if you care about this, this paper is published and it's down here. The modular assembly of polysaccharide degrading marine microbial communities. Okay, so probably I'll try to use maybe 15 minutes, 20 minutes at most to talk about this other project. And so this is pretty much what I just told you. There are degraders and there are consumers. And this is one of the basic things we discovered, right? So it already tells you there's a trophic structure in the system. There's a transfer of energy and materials from the degraders to the secondary consumers. This is not a random network of interactions. Here's a different question that I think is interesting and a really big challenge. Can you predict any of these from genomes? Can you predict anything from genomes? I mean, that's a question. We have hundreds of thousands of genomes sitting there and in servers. What are we doing with that? It's totally unclear. And the reason why we don't know what to do with that is because we don't have the phenotypes, because nobody's growing and characterizing the physiology of these organisms. They're just sequences. And if we had that physiology and phenotypic data, we could do a lot of really cool things. Anyway, that's my little rant. So turns out that no, we cannot tell you much about what the organism does just based on the genomes. I mean, if you look at whether they have the hydrolytic enzymes or not, you get a little bit, you get far in terms of identifying degraders. But what I'm going to talk about is the fact that these secondary consumers come in different flavors, and with genomes you cannot tell their difference. So that's the challenge that we wanted to address, and so we learned how to tell their difference from the genomes. And in particular, what we want to distinguish is organisms that specialize in consuming, and this is you have to pay attention here, it's a subtle difference. They specialize in consuming the metabolic waste products of growth. That means TCA cycle intermediates, perhaps amino acids that leak out of cells and stuff like that, versus the hydrolysis products. Those are the byproducts of the degradation of chitin or any other polysaccharide, meaning the degraders secrete the enzymes. These enzymes diffuse, hit the surface of the material, the polysaccharide in this case. They start chewing, and monosaccharides, monomers, sorry, start to diffuse away from the polymer. Some of them will come back to the degrader. Some will go potentially to other organisms that act as cheaters. People call them cheaters from the social evolution lingo. And so we wanted to see, are these organisms present there? And if so, can we detect them from genomes? So what do we know about chitin degradation? We know quite a bit, but it's mostly from the work done in cholera and other gamma proteobacteria, I think. And so I already told you, I think, basically that there is the enzyme that degrades chitin is called chitinase. This produces oligomers, soluble sugars, and some of them will be imported back to the cell and converted ultimately to fructose 6B, and then that goes to glycolysis. But there is a bunch of other genes that are important, or phenotypes, I should say, that are important to be able to compete effectively in a community that degrades chitin. You can imagine already the question was posed, you have to adhere to the surface of the particle, right? So you need to have perhaps a way to adhere. MSHA pili it's a way to do that. Chitin binding proteins. You have to swim towards the particle. That will really help you if you want to get there in time. So it means that chemotaxis to glucnac, the different forms of oligomer transport, production of antimicrobials to fend off competitors, forming a biofilm, et cetera, et cetera. There's a bunch of different traits that matter and that are not in the canonical pathway. Yeah, so how do we build a collection? How do we build this list of features that we can use to predict who's going to be a good degrader or a good colonizer or not? That's the question. And our approach here was essentially to look at the evolution of the genomes. And so what we did was, and this is something that builds on, by the way, on work I did in my PhD when I was in the Netherlands, we looked at the evolution of chitinases, the enzymes that degrade chitin, across the whole tree of life. This we did with, I don't know if it's written here, but over 8,000 genomes publicly available. And so many of these have chitinases, and then we're able to track down in what part of the tree of life did they lose or gain a chitinase during the history of evolution. And we do that for all gene families that we could identify in these organisms. And then we find cases in which the chitinase, the family of chitinases, the way it evolves is very strongly correlated with the way another gene family evolves. So here I have an example of gene family 1, gene family 2. Gene family 1 could be the chitinase family, and gene family 2 could be anything. And when you look at the tree of life, this is a sketch, you see, in this branch of the tree of life, it looks like the enzyme was lost. In this little branch here, they duplicated. They got it most likely by horizontal gene transfer, another copy. In this other little branch here, seems like all these organisms under this part of the tree have duplicated the enzyme, but this little part here lost it, and so on. You can reconstruct these scenarios. I cannot really go in details about how you do that, but there are ways to do it. And then you can look at all these gene families and ask, are their histories correlated? How correlated are they with the history of the chitinases? So we build fundamentally this network of co-evolution between genes.
By lost, do you mean they become pseudogenes?
No, I mean that the gene wasn't there in the genome. A pseudogene will still be identified the way we did it. Yeah, which is a little issue. I agree. That's a good question, by the way. Yeah, no, we were basically saying, can we find this gene by homology in the genome? Yeah, that's the way to do it.
Gotcha. Thanks.
Yeah, good question. And sorry, by the way, I don't know if this is clear, but in bacteria, there's a huge turnover of genes. In a few generations, they can get new material, new DNA from the environment, and they also lose a lot of genes. There is a bias to lose DNA. So that's why the turnover, in particular, these chitinases evolve extremely fast. Organisms can have up to 15, 20 copies of the chitinases, and some have 5, some have 2, even if they're closely related. Anyway, so we have now this network of coevolution of genes around the chitinases. And the idea is that the things that coevolve very strongly are likely to be functionally linked to the chitinases. And so here's an example of what this looks like, this coevolution. Let's look in detail here. Let's try to, I know that this is you're probably tired by now, but let's focus on this picture here. So this is Shewanella, if I'm not mistaken, the S, but whatever, some taxon, doesn't really matter. These are all relatively close related species. I mean, the same species, they have the Rubidiae species name. And this is a part of their genome. And in yellow, you see the chitinase. That's the enzyme that degrades chitin. In a different part of the genome, quite far away from this locus, where the chitinase is, you see a transcriptional regulator and a chitin binding protein somewhere here. Turns out that when you look at these other guys that are closely related, there was a loss of the chitinase and there was a loss of these two genes. These are independent events because the genes are far away in the genome, but they happened together. And so that's the kind of evidence that we're collecting to build these cases of who interacts with whom. And if you do that for the thousands of genomes we have, you can build these networks. So what we have here is these modules of genes that are all strongly correlated, probably because they form part of an operon or some type of protein complex. So for example, here you have genes that are involved in flagella. I believe these guys here, the GSP are transporters. No, the MSH are the attachment genes. So the out S, I think, is a transporter. Anyway, this is some type of pili down here, the top A and the pil P, and so on. And many, many other things that don't have annotations, by the way. This method is completely independent of annotations. So which makes it actually very powerful. And then we call this set of genes the accessory genes. And then here's another really important detail that requires a little bit of attention. Because we have all these evolutionary events on the tree of life of duplications or losses of genes and so on. We can calculate what is the probability that a chitinase gets lost while the rest of these accessory genes is retained? Or conversely, the probability that any of these accessory genes is lost, but the chitinase is retained. And there are different probabilities for across all the lineages. So we have a distribution of these probabilities, and this is shown here. This is a frequency histogram of the probabilities. And you see the yellow one is the probability that you lose the chitinase, but you retain the accessory machinery. And this distribution is shifted towards the right, meaning that the probabilities are higher here, which means that more often than the opposite case, during the evolution of these organisms, they lose the chitinase, but they retain the machinery. And that's the evolution of a cheater, basically. An organism that has all the equipment to colonize chitin, to grow next to a degrader, to transport the oligosaccharides, to swim towards the particles, etc., but then does not produce the enzyme to degrade chitin. And then here's an example of this. It is Klebsiella pneumoniae. Here on these squares, it looks like Tetris. You have different types of genes and pathways that have co-evolved with the chitinases in this lineage that leads to Klebsiella. Obviously, the glucanate metabolism. Glucanate is a monomer of chitin. Secretion, pillars, blah blah blah, and they all have these things, except that this guy in the middle has lost the chitinase. So that's an example of the evolution of a cheater. We don't call them cheater, we call them exploiters, for whatever historical reasons. Somebody complained in the lab that we were ascribing agency to organisms if we call them cheaters, and then we stuck with exploiters. I know that's much better, but anyway. And so this is what we can do with this approach. We can reconstruct the evolution of these exploiters, and we can identify them. I just have, I think I have maybe five more minutes to 4 p.m. and then we can move to the questions. So as I told you, this is something that we are about to submit, and so we did more. Obviously, now that we have identified these organisms based on genomes, so we developed an approach to identify what is a cheater or exploiter of degraders in this just based on genomes. Now we can try to see what role they play in our communities that I was talking about at the beginning of the talk. And so first thing we did was to say, okay, we have these organisms in the lab. Let's first validate our genomic predictions. Are these exploiters really exploiters? And so we actually developed in the lab a system to study the interactions between the organisms. And this is something that we'll do more with this system. It took a little while to build. But this is a very simple experiment that we did with it. And the system consists on basically, we found a way to dissolve chitin that's not trivial. And we can coat a surface now with this dissolved chitin. The solvent evaporates and you get a beautiful thin transparent sheet of chitin at the bottom of whatever container you have. And then you can see, because it's flat and it's transparent, you can do imaging and see how the bacteria colonize the surface. And some of our organisms are genetically tractable, so we put in, we label them with GFP and RFP. And here's the data. So on the left, you have a degrader. And in gray, you have its growth on chitin when it is in monoculture. And let's look at the other graph. On gray, you have the growth of this exploiter in monoculture, so no growth because an exploiter. So far, so good. But when you combine them, the exploiter grows. It's happy to see this other degrader guy, but the degrader cannot grow as well. It does grow because it needs to start degradation for the exploiter to be able to grow, but its yield is much lower. And this is what we expect from exploiters in general. They are parasites essentially, so they reduce the yield of the degrader. Eventually, they most likely retard the turnover of organic matter in the environment. This is what we think these exploiters are doing. And here's all this information we got from the genomes and our predictions of who's exploiter and who's not exploiter mapped back to that original experiment I showed you at the beginning of the talk with the successions. Here you have, what's on top is a description of the experiment. I already mentioned this, right? The bottles, we take samples over time, we do 16S sequencing, we have culture collections. Now that we know from our culture collection which one is an exploiter and which is a degrader and which is a scavenger of metabolic waste products, we can look at the dynamics of these different groups during the colonization of particles. And in blue, you see the dynamics of a degrader. Okay, in blue, so they come very early, they peak, and then they wane off, they disappear slowly. In the meantime, the black one here are those the waste scavengers that start to slowly creep up until they become relatively abundant. And the yellow one is the exploiter. It's doing pretty much the same thing as a degrader. And this, I think, is because it has all the genetic machinery to chemotax, to attach to chitin, and to do all these things, but it doesn't degrade. So it's just piggybacking on the whatever the degrader is doing. Right, that's maybe what I should say about this, not to delay things much more. And all right, so I promise this is my no, you know what? I'll jump just to this summary I have here, because I think it's already 4 p.m. here in Boston, so I want to give you time, you guys, to ask questions. So all right, so what have we learned? I mean, we have done many other things, but and I've been talking, I think, quite fast. But how can we summarize this whole mess? So I think one of the main take-home messages that I would like you to live with, there's the dog here, is that when you look at the literature, you see what's all these studies of microbiomes, whatever they are, environmental, human-related, whatever. Most of the time, what you see is shopping lists of species, of 16S, OTUs, whatever they are, and there's really no reason to think these are the relevant units of the ecosystem. And so people have mentioned that function is conserved, not taxa, fine. But how do we define what are the functional units? Not clear. Nobody knows how to do this at this point in time. It's an open frontier, something that I think more people should be looking at. For reference, for plant ecology, people, I mean, because it's much more developed and people know a lot about plants, there is a branch of plant ecology, trait-based ecology. People look at plants and they can tell you, they don't care what the species is, but they tell you, there's so much biomass allocated to leaves versus roots, this is the height, and so on. And all these characteristics called traits determine the dynamics of the ecosystem, the function, etc. That's what we would like to do with microbes. It's extremely difficult because we cannot look at a cell and infer these things. So one of the only ways we have to do that is via genomes. So this is a big challenge. Anyway, so this combination of genomics and experiments that I described today, partially at least, what we have learned is a couple of things. First of all, there is a structure of the ecosystem that reflects a trophic structure. That means cascades of energy transfer. Okay, and this is, I think, a general structure of most heterotrophic microbial ecosystems. And if you open an environmental microbiology textbook, you actually will find that picture. Somebody drew it in the 80s or 70s, where they tell you that there's a cascade. For example, in an anaerobic environment, you have the degraders of cellulose and the fermenters and the secondary fermenters and the methanogens. There is a cascade. It's already there. It's just that we didn't know that would happen also in an aerobic system. And it happens, why it happens is actually a very interesting question, but a little bit out of the scope. We can talk about it if you want. What happened here with my slides? So this is what I said before. And then what we have found is that there are at least 2 types of trophic chains. One that is mediated by the metabolic waste of of the species that can grow on the primary substrate, and another one that is mediated by the public goods, by the byproducts of hydrolysis or degradation. And this chemical cascade, in particular in the case of the metabolic waste, creates a number of different niches, actually. This is not something I discuss in any detail here. And so that increases the diversity of the system. You typically see this increasing diversity as the community assembles, and it's because of the niches open all over the place because of the metabolic waste products. They're all slightly different, and there's a lot of opportunity for specialization. And the last thing I can say right here is that these exploiters that we identified shared many genomic features with degraders. And this is what we actually can leverage to make predictions from genomes of who is exploiter and who is not exploiter. And that's something that right now we're extending and trying to do as well with organisms from the human gut or from the rumen microbiome, where there's primarily cellulose degradation. So we would like to identify which organisms are actually degrading cellulose, which are parasitizing the degraders, and which are utilizing fermentation byproducts, for example, in order to build this functional description of the system. Somebody wanted to say something, but before that, let me just say goodbye. No, not goodbye, but say this is the end of my talk, and I want to thank the people here, in particular Tim and Manoshi. Manoshi is now a professor in Indiana, and Shaul Pollack and Matti Gralka are postdocs in my group who did amazing work with exploiters and the genome reconstructions. So, and of course, the funding sources, we have received funding from NSF a couple of times, but our main funding source until now has been the Simons Foundation, which supports an international collaboration of which I'm privileged to be the co-director. And with all these people, it's names that you see down there. All right, so that's all. Now, I guess you guys can shoot with your questions.
Okay, so you know how like the metabolic waste bacteria depend on the oligosaccharide bacteria to collect their whatever they left over to function. Is it possible that it's also reverse dependent? Like if, for example, the waste will choke out the other bacteria, or there's any negative if the other bacteria is not present?
That's a great question. You know, it's possible, and it's something that some of our collaborators are looking at that, and I think we should look at it better. So far, we have not found a very, very compelling case. But I think, so first of all, this product inhibition, I don't think is most likely what's going to happen here, because that's more likely in anaerobic environments. In this case, I don't think you would have to accumulate a really large amount of byproduct. What happens when the byproduct accumulates, by the way, is that they leave the particle. That was part of the answer I couldn't give earlier to one of the questions, Fitnat, I think, of when the byproducts accumulate, then the bacteria leave the particle. And so the residence time is regulated. Anyway, I think that if I can say something interesting about this is that what the interactions will be, you know, we'll measure these things invariantly in the lab, right? To establish if you put them together, do you get a benefit or not? But the point here is that whatever the answer is will depend on how, not only on the microbes, but on the media. And this is something that people tend not to pay attention to. So for instance, if we use our default medium in the lab is a well-defined medium, but it's rich on everything except that it lacks a carbon source, and that's where you put in the chitin, and now you know that, okay, that's a limiting resource. But by definition, this is how people do experiments in general. You have to have one limiting resource. Everything else is in abundance. And so if that's the situation, most likely you will see no interaction other than competition. However, but that's not how the environment works, right? So there are multiple things that are limiting. And so, for example, this is something we're going to start to do now. So I cannot tell you exactly what will happen, but my suspicion is that you will have more of these potential positive interactions that you were implying in your question if we start to remove things. For example, if we remove the vitamins, some of these organisms are auxotroph for vitamins. We know that now. Or if iron is limiting, maybe these exploiters, because they come from different genomic backgrounds, they could bring siderophores or other iron scavenging molecules that are also public goods and will be shared back with the degraders. So those games can start to appear, but if the environment is limiting in multiple dimensions. And so we don't know yet what will happen, but that's kind of my thinking about this issue. More questions about anything?
Are these bacteria taken from a specific region in the ocean, or just not really specific?
No, they are from the coastal ocean, which is typically, there are a lot of nutrients. I mean, for example, there's a lot of ammonia. This is from the coast of Massachusetts, yeah. But there's a lot of mixing. So it's particularly in the place where we collect them, there's a lot of mixing. So I don't think this is necessarily like a bacteria that are native from that environment.
Thank you.
Yeah. Good questions.
Other questions? We have a little bit of time left, so let's make good use of it.
I have a question about the conservation of, well, you were talking about like the genomes that you were pretty much analyzing the genomes of the bacteria. This quarter I was taking a class on viruses, and by chance, did you ever come across any like viral genomes integrated within the bacteria?
Yeah, nice question. Great. Thank you, Claudia. Yeah, so actually not in this data I was showing here. But we have another study we are writing now, where this was a crazy amount of work, and this was done by my dear student Rachel Szabo. So she did play these games of putting the particles in seawater, but this time she sequenced from individual particles. She managed to develop the techniques to do metagenomics. Metagenomics, that means the whole DNA from an individual particle 100 microns in diameter. And turns out, and then we reconstruct the genomes with using, basically you can assemble the genomes, de novo, from the metagenomes. And turns out that, as you were implying, yeah, many of these guys have integrated phages. And well, that's in itself not very surprising, but the fact is that you can see in the metagenomic data, the phages popping out and replicating, because the coverage of the genome is whatever, let's say 10x, and this little piece of DNA where the phages is coverage is 100x. And you can also see that that effect limits the growth of the bacteria. Then you have a lot of noise in the community assembly at the scale of the single particle, and it's, I think, primarily driven by the fact that stochastically, in some cases, the populations explode because of the phages that come out, and in some cases they don't. And that's, to the extent that we can see at the scale of the single particles, the main determinant of how the community develops, how much biomass accumulates, which species are there. It's all about these phages popping up. So it's kind of amazing to me. Yeah, that's a nice question.
So you would say that kind of influences the loss of the, sorry, I can't pronounce the word, the the enzymes that degrade. So would you say like that could possibly influence the loss of that through evolution?
I haven't internalized very well yet what that means for the big picture, but I think what that would do is that you would have a lot of byproducts. I mean, the internal metabolites of the cells that are being blasted are coming out, and this perhaps may be the main way in which these metabolites are released. I mean, in the lab we can see that metabolites leak out of cells even if they're not, exploding. But if they explode, obviously, then you have a lot of resources coming out, and maybe the secondary consumers that don't care about particles, what they're doing is actually eating the pieces of the cells that are being killed by phages. That's likely. Other people have seen similar stuff in other systems. I think it's a very likely thing that is happening all the time in microbiomes. Yeah.
Cool. Thank you.
Yeah, thank you.
Any other questions about the science or about Professor Cordero's path to his current position? I know some people have to leave for classes. That's understandable. If you have to go, that's fine. You're getting a lot of words of appreciation and thanks in the chat box here, Otto.
Oh, thank you. I cannot see the chat, but maybe I stop sharing. Chat. Oh yeah, thank you. Yeah.
And just a word to everybody, if you came in late or if you have to leave early, we are recording this and we'll make a copy of it available. So any last questions? We don't want to hang up Otto any longer than necessary. Because it's late, it's dinner time back in Boston.
Yeah, it's a beautiful afternoon here actually. Anyway, yes.
So, Otto, I was wondering about metabolic fluxes. So it seems like maybe all these measurements somewhat snapshot of what's going on. What do you, what's happening in the temporal scale? Like how far the metabolites are reaching, like distance between bacteria. If you think about the community on those surfaces,
Yeah.
I mean, you have always very elegant experimental design to solve these problems. So I was wondering, what type of approaches are you considering to look at that, nanoscale microenvironments, or what are your thoughts?
Yeah, no, it's a good question because I don't know if you were thinking about this, but if these particles are submerged in an aqueous environment, you would think most of the metabolites, whatever they are lost, right? So we haven't looked at that too much in the context of this metabolic byproducts like of growth, the waste products. Other collaborators have looked at that and made models of it and they think it works. But we have looked at this in the context of the hydrolysis products, the oligosaccharides. And what we found was that if the cells that degrade, let's imagine, the scenario here is only one degrader, no community, just one species. And if these cells are just uniformly scattered around the surface, each of them has the ability to degrade the particle, but they won't be able to grow because 99.9% of the degradation byproducts that the enzymes release are going to diffuse away. And the number is I didn't make that up. It's more than 99%. So the solution to that problem is for the cells to cluster and to form little aggregates. I'm talking about clusters of like 20 cells in diameters, tiny little clusters, and which is 20 microns. And then the cells can grow. And this is something we discovered basically first when we were doing simulations of this process. And we realized that if you make the polymer, the material really hard to degrade, then you will only get growth if they form these clusters in our individual based models. And later we did experiments and we found exactly the same thing. So at least that's one thing that I can say regarding the diffusion of metabolites in space is that at least in the context of the oligosaccharides, the degradation byproducts, you get the emergence of these cooperative cell clusters because they need to actually be next to each other to get the byproducts from each other and grow.
Yeah, very nice. Thank you. No, that makes sense. Yeah,
I'm glad it does.
Great, thank you. Great questions, everyone. Great talk, Otto. Thank you so much.
Thank you. It was really fun. And
really enjoyed having you here.
I hope I can meet you, all of you, in person at some point next year, hopefully.
Hopefully, maybe we can bring you out for BD2K summer.
I would love that.
Yeah, that would be great.
All right.
So thank you very much. Just a quick reminder, everyone, we'll be having 2 additional Pathways to Careers in Genomics talks in winter and spring quarter. Brandon Obunu from Yale University and Carolina Reyes, who is a research scientist in Switzerland right now, working in private industry. And we'll be announcing dates soon. So if you want to get on our mailing list, reach out to me or Sandra. And we'll be sure to send you that announcement. You can also follow us on Facebook and the Genomics Institute Facebook and Instagram pages as well. So once again, thank you so much.
Yeah, thank you.
It's been really great having you here. Thank you everybody for turning out today.